Oxygen that killed life. The paradox of the vital gas. When we look out at the world today, we see a planet defined by the presence of oxygen. It is the invisible medium through which modern biology moves, the fuel for the metabolic fire that burns within every animal, every bird, and every human being. We view oxygen as synonymous with life itself. In our search for extraterrestrial intelligence or habitable worlds orbiting distant stars, the spectroscopic signature of oxygen is the holy grail, the definitive marker that suggests a biosphere is active and thriving. We are so dependent on this diatomic molecule that a deprivation of it for mere minutes results in permanent brain damage or death. It is the breath of life, the sustaining fluid of the biosphere, and the chemical key that unlocked the evolution of complex multicellular organisms. Yet, this perspective is a bias born of our specific place in time. There is a dark history to this element, a chapter in the planetary biography where oxygen was not the nurturer of life, but its greatest destroyer. To the ancient inhabitants of the early Earth, oxygen was not a breath of fresh air. It was a terrifying, corrosive poison, a chemical weapon of mass destruction that unleashed a global holocaust, wiping out the vast majority of living things and freezing the planet into a ball of ice for millions of years. This event, known to science as the Great Oxidation Event, stands as the most significant environmental catastrophe in the history of our world. It is the story of how a single metabolic innovation by a microscopic organism nearly ended the experiment of life on Earth before it had truly begun. The alien Earth. To understand the magnitude of this catastrophe, one must first strip away the Earth as we know it. Travel back in time approximately 3 billion years, and you would find yourself on a planet that is unrecognizable, an alien world orbiting the familiar sun. The sky above is not the deep, comforting blue we enjoy today, but likely a hazy, terrified orange or pink, similar to the atmosphere of Titan, Saturn's moon. This color comes from a thick smog of methane and hydrocarbon particles suspended in the upper atmosphere. The sun itself is different, appearing slightly smaller and shining with only about 70% of its modern luminosity. This is the time of the faint young sun paradox, a period when the solar output was so weak that the Earth should have been a frozen wasteland, yet geological evidence confirms the presence of liquid water. The planet remained warm thanks to a potent greenhouse effect, driven not by carbon dioxide alone, but by methane, a gas with a warming potential nearly 30 times that of CO2. The oceans of this Archean eon were not the clear blue expanses of the modern world. They were green, rich in dissolved iron, or perhaps purple, teeming with microbial mats that utilized different pigments to harvest the dim sunlight. But the most striking difference was the air. There was no free oxygen. The atmosphere was a reducing one, composed of nitrogen, carbon dioxide, methane, and perhaps traces of hydrogen and ammonia. In this anoxic world, life flourished. It was a simple life, microscopic and single-celled, but it was diverse and robust. These organisms were the anaerobes. They had evolved distinct metabolic pathways to generate energy without the need for oxygen. Some were methanogens, ancient archaea that consumed hydrogen and carbon dioxide to produce methane, thereby maintaining the planetary thermostat. Others utilized sulfur or iron in their metabolic cycles. For hundreds of millions of years, these organisms ruled the Earth, thriving in the hydrothermal vents, the muds of the shallow seas, and the deep subsurface. They had no defense against oxygen because there was no need for one. To them, oxygen was a high-energy radical, a chemical that would steal electrons from their delicate organic structures, breaking apart the proteins and DNA that constituted their being. They lived in a Garden of Eden that was strictly anaerobic, the green revolutionaries. The agent of destruction arrived quietly, born from a random mutation and the relentless pressure of natural selection. Somewhere in the shallow, sunlit waters of the ancient ocean, a new type of bacterium evolved. These were the ancestors of modern cyanobacteria. Life had already discovered the trick of photosynthesis, the ability to turn sunlight into chemical energy. But the early versions were anoxygenic. Bacteria like the purple sulfur bacteria used hydrogen sulfide or dissolved iron as electron donors to drive the fixation of carbon. This process was efficient enough for the time, but it was limited by the availability of these chemical resources. Hydrogen sulfide is found only in specific volcanic environments. It is not ubiquitous. The evolutionary leap made by the cyanobacteria was the development of oxygenic photosynthesis. They engineered a molecular machine of staggering complexity, known today as photosystem II. This biological apparatus had the power to do something chemically difficult. It could split water. Water is everywhere. By using the energy of sunlight to rip electrons from water molecules, these bacteria gained access to an unlimited supply of hydrogen for their metabolic needs. The waste product of this reaction, dumped into the environment because it was of no use to the bacterium, was a highly reactive toxic gas, molecular oxygen. For a long time, this new waste product did not cause a global crisis. The Earth had vast chemical buffers. The oceans were filled with dissolved iron, a legacy of the planet's volcanic youth. As the cyanobacteria pumped out oxygen, the gas immediately reacted with the iron, turning it into rust. This rust settled onto the ocean floor in massive layers, creating the banded iron formations that we mine today for steel. These geological strata are the silent graveyards of the oxygen produced during millions of years of whiffs and starts. The planet was soaking up the poison like a sponge. The oxygen reacted with sulfur, with hydrogen, and with the rock itself. The cyanobacteria were prolific, forming vast mats called stromatolites, layered structures of mucus and sediment that grew upward toward the sun. They were successful because they had tapped into an infinite resource, water. But for hundreds of millions of years, their dangerous exhaust was contained, the tipping point. Eventually, the sponge became saturated. The sinks were full. The dissolved iron in the oceans precipitated out completely, leaving the waters clear for the first time. The volcanic gases that had previously scrubbed oxygen from the air began to diminish as the Earth's tectonic activity stabilized. Around 2.4 billion years ago, the balance shifted. The production of oxygen by the relentless biological machinery of the cyanobacteria finally outpaced the planet's ability to absorb it. The gas began to accumulate in the water, and then it bubbled up into the atmosphere. This moment marks the onset of the Great Oxidation Event. In the geological blink of an eye, the chemistry of the entire surface of the Earth was rewritten. The concentration of oxygen rose from less than 1 part per million to perhaps 1 or 2% of the atmosphere. By modern standards, this is unbreathable, but to the anaerobic biosphere of the time, it was an environmental apocalypse. The holocaust of the anaerobes. We often speak of extinction events in terms of large animals, the dinosaurs, the mammoths, the trilobites. But the Great Oxidation Event was a mass extinction of the invisible. It was a microbial holocaust. For the dominant life forms on Earth, the air had suddenly turned into acid. Oxygen is a powerful oxidant. It aggressively strips electrons from other molecules. When it encounters organic matter that has not evolved specific enzymatic defenses, it creates reactive oxygen species, superoxide radicals, hydrogen peroxide, and hydroxyl radicals. These chemical fragments act like shrapnel within a cell. They attack the lipid membranes, causing them to leak. They shatter the enzymatic machinery required for metabolism. They break the double helix of DNA, causing lethal mutations. The anaerobes died in numbers that are impossible to quantify. Entire ecosystems that had thrived for 1 billion years were wiped out. The microbial mats that carpeted the seafloors bleached and decayed. The open ocean, once a haven for anaerobic swimmers, became a kill zone. The only survivors were those that managed to retreat into the deep, dark recesses of the planet where oxygen could not reach, into the mud of the abyssal plains, into the crust of the earth, or into the gut environments of larger organisms that would evolve much later. Even today, the descendants of these survivors, such as the bacteria that cause botulism or gangrene, are obligate anaerobes. Oxygen is still lethal to them. They are the refugees of a war lost 2 billion years ago, hiding in the shadows of an oxygenated world. The cyanobacteria, the architects of this destruction, were not immune to their own poison. They too had to evolve mechanisms to deal with the toxic byproduct of their success. They developed antioxidant enzymes like catalase and superoxide dismutase, molecular shields that could neutralize the reactive oxygen species before they did fatal damage. This was an arms race against their own waste, a struggle to adapt to the very environment they were creating. The climatic collapse. The biological devastation was matched by a climatological catastrophe. As oxygen flooded the atmosphere, it encountered the methane haze that had kept the Earth warm. Methane is unstable in an oxygenated atmosphere. The two react to form carbon dioxide and water. While carbon dioxide is a greenhouse gas, it is far less potent than methane. As the oxygen scrubbed the methane from the sky, the planetary blanket was ripped away. The greenhouse effect collapsed. The Earth plunged into a deep freeze. This was not a standard ice age of the kind that has affected humans in the recent past. This was the Huronian glaciation, a global event so severe that it may have resulted in a snowball Earth. The temperatures plummeted, and ice sheets spread from the poles to the equator. The entire planet may have been encased in a shell of ice kilometers thick, with the oceans sealed off from the atmosphere. The hydrological cycle slowed to a crawl. Photosynthesis would have been severely curtailed as the ice blocked the sunlight, creating a feedback loop that nearly extinguished the very cyanobacteria that had started the process. Life hung by a thread. For millions of years, the Earth was a white, frozen ball drifting through space. The few pockets of liquid water near volcanic vents or beneath thin patches of ice became the last refuges for the survivors. The oxygen that had killed the anaerobes had now indirectly frozen the world, threatening to end the biological experiment entirely. It was only the slow, grinding mechanics of plate tectonics that saved the planet. Volcanoes continued to erupt beneath the ice, pumping carbon dioxide into the atmosphere. With no liquid rain to wash the CO2 out of the air and weather the rocks, the gas accumulated over millions of years until the greenhouse effect was strong enough to melt the ice. The thaw, when it finally came, was likely violent and rapid, leading to a hothouse Earth before the climate stabilized. The long pause. Following the chaos of the Great Oxidation Event and the Huronian glaciation, the Earth entered a period often referred to by geologists as the, quote, boring billion. For roughly 1 billion years, from 1.8 billion to 800 million years ago, the environment and the biosphere seemed to stagnate. Oxygen levels stabilized, but they remained low, perhaps around 1 to 10% of modern levels. The deep oceans remained anoxic and sulfidic, a condition known as the Canfield Ocean. However, this period was not truly boring. It was a time of hidden alchemy. The presence of oxygen, even at low levels, had fundamentally changed the energetic possibilities of life. Anaerobic respiration, fermentation, is inefficient. It extracts only a fraction of the energy potential stored in a glucose molecule. Aerobic respiration, which uses oxygen to burn fuel, is vastly more powerful. generating nearly 20 times more ATP, adenosine triphosphate, per unit of fuel. This massive jump in available energy was the prerequisite for complexity. It was during this long, quiet interval that the most important merger in the history of life occurred. A predatory or scavenging archaean cell engulfed a smaller bacterium, likely a purple non-sulfur bacterium that had evolved to detoxify oxygen and perhaps generate energy from it. Instead of digesting the meal, the host cell and the captive entered into a symbiotic relationship. The internal bacterium provided the host with ATP derived from oxygen, protecting the host from toxicity and powering its growth. In exchange, the host provided protection and nutrients. Over time, this internal bacterium lost its independence, stripping down its genome to the bare essentials, and became the mitochondrion. This was the birth of the eukaryote, the complex cell with a nucleus and organelles. Every plant, animal, fungus, and protist alive today is a descendant of this ancient merger. The toxic gas that had slaughtered the biosphere had forced life to innovate. By harnessing the high-energy reactivity of oxygen, life broke through the glass ceiling of complexity. The very danger of oxygen, its frantic desire for electrons, was harnessed to drive the turbines of the mitochondria. The poison became the potion. The second rise. The story of oxygen does not end with the Great Oxidation Event. A second major pulse of oxygenation occurred much later, during the Neoproterozoic era, roughly 800 to 500 million years ago. The causes of this second rise are still debated, possibly linked to the breakup of supercontinents and the burial of vast amounts of organic carbon, which prevented it from reacting with oxygen. This second rise pushed oxygen levels closer to modern standards. It was this second surge that paved the way for animals. The high metabolic demands of predation, of muscle movement, of nervous systems, and of large body sizes require the high-octane fuel that is oxygen. The Cambrian explosion, the sudden appearance of most major animal phyla in the fossil record, coincides with this rise in oxygen. The relationship is causal. Without the atmospheric abundance of O2, the energetic constraints on biology would have kept life microscopic and sluggish. The predators that swam in the Cambrian seas, the armored trilobites, and the worm-like ancestors of vertebrates were all powered by the chemical fire ignited by oxygen, the dual nature of oxygen. Reflecting on the Great Oxidation Event forces us to confront the dual nature of oxygen. It is a creator and a destroyer. It is the reason we exist, yet it causes the molecular damage that leads to aging and cancer. Every time we take a breath, we are keeping ourselves alive, but we are also generating the free radicals that slowly degrade our bodies over time. We are obligate aerobes, addicted to a toxic substance that allows us to live fast and bright, rather than slow and eternal. The event also teaches a humbling lesson about the power of life to shape the planet. We often think of geology as the stage upon which biology acts, but the Great Oxidation Event proves that biology can rewrite the script of geology. Microscopic bacteria, working blindly and without intent, altered the chemical composition of the entire atmosphere, rusted the oceans, and froze the world. They destroyed their own environment through their success. This parallels the modern human situation. Like the cyanobacteria, we have discovered a powerful source of energy, fossil fuels, and our use of it is altering the atmospheric composition, driving a greenhouse effect that threatens our own civilization. The cyanobacteria had no capacity to understand what they were doing. They were simple machines of reproduction. We, however, possess the hindsight of billions of years. We know the story of the oxygen catastrophe. We know that rapid environmental change can lead to mass extinction. The legacy of the rust. The legacy of the Great Oxidation Event is written in the rocks. If you travel to the Pilbara Craton in Australia or the Canadian Shield, you can see the banded iron formations, the beautiful red striped rocks that testify to the rusting of the ancient world. These formations are not just geological curiosities. They are the foundation of our industrial civilization. The steel in our cars, our skyscrapers, and our ships comes from the iron precipitated out of the ocean 2.5 billion years ago by the first breaths of cyanobacteria. We are building our future on the wreckage of the first apocalypse. Furthermore, the ozone layer, which protects the Earth from lethal ultraviolet radiation, is a direct product of the oxygenated atmosphere. High in the stratosphere, solar radiation splits O2 molecules, allowing them to recombine as ozone O3. Without this shield, the surface of the land would have remained sterile, bombarded by DNA-shattering UV rays. The colonization of the land by plants and animals was only possible because of the protective canopy built by oxygen. Conclusion: the title "Oxygen That Killed Life" is not hyperbole. It is a historical fact. The transition from an anaerobic to an aerobic world was the most violent disruption in the history of terrestrial life. It destroyed a stable billion-year-old order. It was a chemical warfare waged by the future against the past. Yet from that wreckage emerged the complexity that defines our world. The Great Oxidation Event serves as a reminder that stability in nature is an illusion. The Earth is a dynamic system, constantly seeking equilibrium, but rarely finding it for long. Life is resilient, but it is also fragile in the face of rapid change. The oxygen that fills our lungs today is a ghost of a catastrophic past, a token of the time when the sky turned blue and the world froze, paving the way for the unlikely rise of sentient beings who could look back and tell the story. The chemical mechanism of the Great Dying. To truly appreciate the mechanism of the slaughter, we must delve deeper into the biochemistry of the pre-oxygen cell. The ancient anaerobes utilized metalloenzymes that were fundamentally incompatible with oxygen. These enzymes, often containing iron-sulfur clusters, were the workhorses of anaerobic metabolism. They facilitated the transfer of electrons, the fixation of nitrogen, and the synthesis of amino acids. Oxygen, with its high electronegativity, is a bully. When it enters a cell containing these exposed iron-sulfur clusters, it does not wait to be invited into a reaction. It attacks the iron, oxidizing it. This oxidation destroys the cluster, rendering the enzyme useless. The cell's metabolic machinery grinds to a halt. It is akin to pouring sand into an engine. The moving parts seize up. Furthermore, the interaction of oxygen with other cellular components generates the reactive oxygen species mentioned earlier. The hydroxyl radical, in particular, is devastating. It reacts at diffusion-limited rates, meaning it reacts with the first thing it hits. There is no targeting, no specificity. It simply tears electrons off the nearest molecule. If that molecule happens to be part of the DNA backbone, the genetic code is severed. If it is a lipid in the cell membrane, a chain reaction of lipid peroxidation begins, compromising the barrier that holds the cell together. The anaerobic cell, lacking the sophisticated repair mechanisms and antioxidant enzymes of later life, essentially disintegrates from the inside out. The deep Earth response. The geological response to the oxygenation was not limited to the surface. The chemistry of the deep Earth was also affected. As oxygenated water eventually circulated into the crust and interacted with subducting plates, it changed the oxidation state of the mantle wedge. This, in turn, affected the types of minerals that crystallized from magmas. The mineral evolution of Earth exploded in diversity. Robert Hazen and other mineralogists have estimated that 2/3 of the known mineral species on Earth owe their existence to the presence of biological oxygen. Minerals like turquoise, malachite, and azurite are oxidized forms of copper that would not exist on a strictly anaerobic planet. The Great Oxidation Event did not just kill life, it painted the rocks. It created a colorful Earth, replacing the drab grays and blacks of the Archean basalts with the vibrant reds, greens, and blues of oxidized minerals. The ozone shield and the march to land. Before the Great Oxidation Event, the land surfaces of Earth were stark and barren, more like the surface of Mars than the Earth we know. Ultraviolet radiation from the sun sterilized the continents. Life was strictly confined to the water, which provided a natural shield against UV rays. The accumulation of oxygen in the atmosphere led to the formation of the ozone layer. Ozone is a triatomic molecule of oxygen that is particularly good at absorbing UVB and UVC radiation. As the ozone layer thickened, the lethal flux of radiation reaching the surface diminished. This opened up a new frontier. Microbial mats were the first to colonize the wet margins of the land, followed eventually by plants and arthropods. This colonization triggered another feedback loop. Land plants, with their roots penetrating the rocks, accelerated chemical weathering. This weathering pulled carbon dioxide out of the atmosphere and washed nutrients like phosphorus into the oceans, fueling further blooms of oxygen-producing marine life. The conquest of land, made possible by the toxic waste of the cyanobacteria, eventually led to the forests that cover the continents today, acting as the planet's second lung. The future of oxygen. As we look to the future, we must realize that the oxygenated atmosphere is not a permanent feature of the Earth. It is a state of disequilibrium maintained by the constant effort of the biosphere. In the far distant future, as the sun continues to brighten as part of its stellar evolution, the Earth will eventually become too hot for liquid water. The weathering of rocks will accelerate, scrubbing all carbon dioxide from the air. Without CO2, photosynthesis will cease. The plants will die, and with them, the supply of oxygen will be cut off. The oxygen that remains will slowly react with the surface rocks and disappear. The Earth will return to an anoxic state, perhaps dominated once again by simple, anaerobic microbes, before the planet becomes completely sterilized by the dying sun. The era of oxygen, the era of complex life, is a temporary window in the deep time of the cosmos. The lesson of the oxygen that killed life is one of profound interconnectedness. A single molecular innovation in a single bacterium changed the color of the sky, the chemistry of the oceans, the composition of the rocks, and the destiny of all biological evolution. It teaches us that the biosphere is not a passive passenger on the planet, but a driver of planetary evolution. It warns us that waste products, if allowed to accumulate, can induce catastrophic phase shifts. And it reminds us that out of the greatest destruction can come the greatest complexity. The oxygen catastrophe was the crucible in which the modern world was forged. We are the children of the survivors, breathing the very gas that once threatened to extinguish the flame of life forever. The snowball Earth hypothesis detail. The connection between the Great Oxidation Event and the snowball Earth, specifically the Huronian glaciation, deserves closer scrutiny as a key component of the killing. The mechanics of this freeze were terrifyingly efficient. The pre-GOE atmosphere was likely sustained by a methane concentration of 100 to 10,000 parts per million. Methane is produced by methanogens, which thrive in anaerobic conditions. As oxygen levels rose, it didn't just poison the methanogens directly. It reacted with the methane in the atmosphere. The chemical reaction CH4 plus 2O2 to CO2 plus 2H2O essentially swapped a blanket of heavy wool, methane, for a thin sheet of cotton, CO2. The solar luminosity was roughly 80 to 85% of present levels. Without the methane greenhouse, the equilibrium temperature of the Earth dropped well below the freezing point of water. Once the ice began to form at the poles, the albedo effect took over. Ice reflects sunlight. The more ice formed, the more sunlight was reflected back into space, cooling the planet further and creating more ice. This runaway positive feedback loop continued until the ice sheets met at the equator. The global temperature may have dropped to minus 50 degrees Celsius on average. The oceans were covered in ice up to a kilometer thick. This choked off the exchange of gases between the ocean and atmosphere. The darkness beneath the ice caused a collapse in photosynthetic activity, nearly driving the cyanobacteria themselves to extinction. It was a near-suicide pact by the oxygen producers. The life that survived did so in isolated pockets, perhaps in the slush near the equator, or around geothermal vents where heat from the Earth kept water liquid. The evolutionary bottleneck created by this freeze was severe. It pruned the tree of life, leaving only the most resilient branches, the recovery and the boring billion. The recovery from the snowball Earth was equally dramatic. With the silicate weathering cycle halted, since rocks were covered in ice, volcanic CO2 built up in the atmosphere until it reached incredibly high levels, perhaps 10% of the atmosphere. This created a super greenhouse effect that melted the ice rapidly. The Earth swung from a freezer to a sauna. Massive cap carbonates found in the geological record attest to the rapid precipitation of limestone in the aftermath of the melt. Following this turmoil, the Earth settled into the boring billion, or the Mesoproterozoic era. The term boring refers to the relative lack of dramatic perturbations in the carbon isotope record, but biologically, it was the incubation period for eukaryotes. The ocean chemistry was stratified, an oxygenated surface layer where cyanobacteria lived, and a sulfitic anoxic deep layer, the Canfield Ocean. This sulfitic condition was toxic to many eukaryotes, which may explain why the evolution of complex multicellular life took so long to get started. The sulfide toxicity acted as a brake on evolution, keeping life simple and small for 1 billion years. It was only when the second oxygenation event cleared the sulfide and oxygenated the deep ocean that large animals could evolve. Thus, oxygen played the role of both the executioner, during the GOE, and the liberator, during the Neoproterozoic, the isotope evidence. How do we know this story is true? The evidence lies in the atomic weight of sulfur. Sulfur has 4 stable isotopes: 32s, 33s, 34s, and 36s. In an atmosphere without oxygen, ultraviolet light interacts with sulfur dioxide to produce specific ratios of these isotopes, a phenomenon known as mass-independent fractionation, MIF. This MIF signal is preserved in Archean rocks. It acts as a fingerprint of an oxygen-free atmosphere. Around 2.4 billion years ago, this MIF signal vanishes from the rock record completely. This disappearance is the smoking gun. It indicates that an ozone layer had formed, blocking the UV light that drives the fractionation, and that atmospheric oxygen levels had risen above 10 to the power of minus 5% atmospheric levels. This geochemical signal is one of the most robust pieces of evidence in all of Earth science, anchoring the narrative of the Great Oxidation Event in hard data. The role of nickel. Recent scientific detective work has added another layer to the story, explaining why the event happened when it did. Why didn't oxygen rise earlier? Cyanobacteria likely evolved hundreds of millions of years before the GOE. One hypothesis involves nickel. Methanogens, the bacteria producing the methane greenhouse, require nickel as a cofactor for their enzymes. Analysis of ancient rocks suggests that the supply of volcanic nickel to the oceans dropped significantly around 2.7 to 2.5 billion years ago as the Earth's mantle cooled. This nickel famine starved the methanogens. With their population crashing, methane production slowed. This reduced the sink for oxygen since methane reacts with oxygen. The decline of the methanogens loosened the chokehold on the atmosphere, allowing the oxygen produced by cyanobacteria to finally build up. Thus, a subtle change in the composition of volcanic lava may have tipped the scales in favor of the oxygen revolution. The biological innovation of the oxygen-evolving complex. At the heart of the catastrophe lies a specific cluster of atoms, the manganese cluster, Mn4CaO5. This is the catalytic core of photosystem II. It is the only biological structure in the known universe capable of oxidizing water at ambient temperatures. The evolution of this cluster is an evolutionary marvel. It likely evolved from simpler manganese oxidizing enzymes. The structure acts like a capacitor, storing the energy of 4 photons to strip 4 electrons and 4 protons from 2 water molecules, releasing 1 molecule of O2. This mechanism is so difficult that it seemingly evolved only once. Every plant, algae, and cyanobacterium on Earth inherited this single invention. There is no alternative pathway for oxygenic photosynthesis. This suggests that the Great Oxidation event was not inevitable. It was contingent on a specific, rare evolutionary accident. Had the manganese cluster not evolved, Earth might still be a methane-hazed, slime-covered world dominated by anaerobes. Comparison with other planets. This contingency has profound implications for astrobiology. We assume that if we find a planet in the habitable zone, it will eventually develop an oxygen atmosphere. But the history of Earth suggests that oxygenation is a difficult, dangerous, and protracted process. It requires the invention of water splitting, the overcoming of reductant sinks, and the survival of the subsequent glaciation. Many planets might get stuck in the Archean phase, teeming with anaerobic microbial life, but never making the leap to high-energy oxygen atmospheres. The oxygen that killed life might be a rare filter in the cosmos, a hurdle that few biospheres manage to clear. The human parallels and responsibility. The narrative of the Great Oxidation event resonates with the Anthropocene. We are currently engaging in a global geoengineering experiment. By releasing gigatons of carbon dioxide, we are reversing the cooling trend of the last few million years. Just as the cyanobacteria pushed the Earth out of equilibrium, we are doing the same. The difference lies in cognition. The cyanobacteria were mindless. We are not. We have the capacity to measure the changes, predict the outcomes, and mitigate the damage. The Great Oxidation event serves as a memento mori for civilizations. Planetary atmospheres are fragile, and life is a potent geological force. The oxygen that grants us life is a reminder of the deadliness of unchecked biological expansion. Final summary of the mechanism. To summarize the sequence of the oxygen that killed life. 1. The Archean Earth was anoxic, warm, and methane-rich, populated by anaerobic microbes. 2. Cyanobacteria evolved oxygenic photosynthesis, splitting water and releasing O2 as waste. 3. For millions of years, O2 was absorbed by chemical sinks, iron, sulfur, volcanic gases. 4. A decline in nickel-starved methanogens, reducing the methane counterforce. 5. The sinks filled up. O2 accumulated in the atmosphere, the Great Oxidation event. 6. O2, toxic to anaerobes, caused a mass extinction, the microbial holocaust. 7. O2 reacted with methane, destroying the greenhouse effect. 8. The Earth froze. Huronian glaciation/snowball Earth. 9. Volcanic CO2 eventually melted the ice. 10. Life adapted via endosymbiosis, mitochondria, and protective enzymes, leading to the evolution of complex eukaryotes. This sequence represents the most significant transformation of the Earth's surface environment since its formation. It established the chemical dichotomy of the modern world, an oxidized surface and a reduced interior. It set the stage for the coevolution of the biosphere and the geosphere, where life regulates the environment that in turn shapes life. The invisible boundary. There is a layer in the rock record, invisible to the naked eye but screaming to the mass spectrometer, that marks the boundary between the old world and the new. Below it, the sulfur isotopes vary wildly. Above it, they track together. That thin line represents the death of the old order. It represents the moment the sky cleared and the lethal UV rays were stopped by the ozone. It represents the billions of microscopic deaths that paved the way for our existence. We walk on a planet shaped by this ancient catastrophe. The oxygen in our blood is the legacy of the survivors. The story of the oxygen that killed life is, ultimately, the story of how we came to be. It is a narrative of destruction and rebirth, a testament to the resilience of life, and a warning about the power of metabolic waste to reshape worlds. The physiological cost of oxygen. Even today, the toxicity of oxygen dictates our biology. Our cells are under constant siege. The electron transport chain in our mitochondria is imperfect. It leaks electrons, which react with oxygen to form superoxide. Our bodies spend a significant amount of energy maintaining a defense system of antioxidants, vitamin C, vitamin E, glutathione. The diseases of old age, Alzheimer's, Parkinson's, cardiovascular disease, are partly linked to the cumulative damage of oxidative stress. We are slowly rusting from the inside. We pay for the high energy of aerobic life with the gradual degradation of our tissues. The oxygen that killed life never really stopped killing. It just kills us slowly enough that we have time to reproduce first. This is the Faustian bargain of aerobic life. We traded longevity and stability for energy and complexity. The anaerobes that hide in the mud might live unchanged for eons, but they will never build a civilization, never write a poem, and never look up at the stars. The oxygen catastrophe gave us the power to dream at the cost of our own mortality. Evolutionary stagnation versus radiations. The long periods of stability, like the boring billion, contrast sharply with the rapid changes of the GOE and the later Cambrian explosion. It seems that stress and catastrophe are the drivers of evolutionary innovation. When the environment is stable, life optimizes. When the environment crashes, life innovates. The Great Oxidation Event was the ultimate crash. It forced the integration of different lineages, endosymbiosis. It forced the development of hard shells, later as oxygen allowed for collagen synthesis. It forced the development of complex regulatory networks. The killer oxygen was the spur that drove life out of the microbial slime and toward the infinite forms most beautiful and most wonderful that Darwin described. The future geological record. If an alien geologist were to visit Earth 1 billion years from now, they would see the distinct strata, the Archean basalts, the banded iron formations marking the transition, the carbonates of the post-glacial thaw, and the oxidized red beds of the Proterozoic. They would read the history of a planet that was terraformed by its own inhabitants. They might also see a new layer, the Anthropocene layer, marked by plastics, radionuclides, and a spike in CO2. They would wonder if this second great disruption led to another collapse or another leap in complexity. The history of the Great Oxidation Event suggests that life is hard to kill, but civilization is fragile. The oxygen crisis took millions of years to resolve. We do not have that kind of time. In conclusion, the story of the oxygen that killed life is the foundational myth of modern biology, not a myth in the sense of a falsehood, but in the sense of a grand explanatory narrative. It defines the parameters of our existence. It explains why the sky is blue, why our blood is red, hemoglobin requires oxygen, why we need to eat so much to fuel aerobic metabolism, and why we die. It is a story of cosmic improvisation, where a waste product became the elixir of life and a global poison became the medium of consciousness. The complexity of the nitrogen cycle. To add further depth, one must consider the nitrogen cycle. In the anaerobic world, nitrogen fixation, turning N2 gas into usable ammonia, was difficult but not hindered by oxygen. The enzyme nitrogenase, which performs this feat, is destroyed by oxygen. When the great oxidation event occurred, the nitrogen cycle was thrown into chaos. Nitrogen fixers had to evolve specialized cells, heterocysts, or temporal separation, fixing nitrogen only at night, to protect their nitrogenase from oxygen. This added another layer of complexity to the microbial world. The crisis of oxygen was not just about respiration. It disrupted the building blocks of proteins and DNA by cutting off the nitrogen supply until life adapted. This underscores the totalizing nature of the event. It wasn't just the air, it was the nutrients, the temperature, and the light. Everything changed, the role of plate tectonics in the balance. Geology and biology danced a tango during this era. The burial of organic carbon is crucial for oxygen buildup. If organic matter, dead bacteria, decays, it consumes oxygen. If it is buried in sediment before it can decay, the oxygen remains in the atmosphere. The rate of burial depends on plate tectonics, erosion rates, and the formation of sedimentary basins. The Great Oxidation Event likely coincided with a period of increased tectonic activity that facilitated the burial of vast amounts of carbon. This geological assistance was necessary to tip the scales against the chemical sinks. It shows that life does not exist in a vacuum. It is intimately tied to the grinding of the continental plates, the search for life elsewhere. Finally, as we build telescopes like the James Webb Space Telescope and the future Habitable Worlds Observatory, the lesson of the GOE guides our eye. We are not just looking for oxygen. We are looking for the disequilibrium of oxygen and methane together. Seeing oxygen alone might be a false positive produced by UV splitting water on a dead planet. But seeing oxygen alongside methane, two gases that should destroy each other, tells us that something is actively replenishing them, that something is life. The chemical signature of the war between the methanogens and the cyanobacteria, the war that nearly killed the Earth, is the very beacon that might reveal we are not alone. The oxygen that killed life is the lighthouse of the universe. This comprehensive overview covers the geochemical, biological, evolutionary, and climatological aspects of the Great Oxidation Event, providing a detailed narrative of how the waste product of a microbe transformed the planet from a reducing atmosphere to an oxidizing one, causing mass extinction, global glaciation, and ultimately paving the way for complex life. The story serves as a powerful testament to the transformative power of biological processes on a planetary scale.